Welcome to the seminar about CRISPR technology. I am Erin, and this is my colleague George, and we'll be talking about CRISPR technology in agriculture and food industry. We'll be looking at the regulation of genetically modified organisms, or GMOs, and how this relates to CRISPR and where CRISPR fits into that framework.
All right, so the engineering of food really began with the invention of agriculture, and the Neolithic Revolution marked the moment where humans abandoned the hunter-gatherer lifestyle and embraced the idea of domesticating nature for their benefit. And in farming, we had selective breeding, which allowed us to choose the desirable traits we want in our food, such as nutrition, taste, resistance to disease. And maize is probably one of the best examples of this when you consider that its ancestor, Usyd, was just produced a very tiny ear, and then this was selectively bred to form one of the world's most important crops. All right, so after about hundreds of thousands of years of just traditional agriculture, we arrived at a new method of quickly providing us the traits of interest thanks to advances in genetics and molecular biology. And this involved incorporating DNA into organisms, and these transgenic products came to be known as genetically modified organisms, or GMOs. And although they achieved success in many ways, they faced a negative public perception based on safety concerns, as well as some environmental concerns, such as gene transfer to non-target species or reducing the effectiveness of the available pesticides.
Okay, so in the research papers that we'll be looking at CRISPR knocking out a certain gene of interest and making that gene, impeding the function of that gene. The gRNA the gRNA has a particular sequence that recognizes the DNA that we'll be looking at, and it forms a complex with Cas9. That complex then comes into contact with the DNA, forms a double-stranded break, and then non-homologous end joining is used to repair that break. Non-homologous end joining can then cause insertion or deletion mutations that can cause frame shift and therefore knock out the function of that gene. GMOs have had a very bad light has been framed on GMOs, and this is really weird because GMOs form up 90% of the large-scale crops in the USA. These include soy, cotton, canola, corn, and sugar beets. While in over 60 countries like Australia, Japan, and many of the European countries, GMO crops are highly regulated and for both production and for distribution. Many of these countries, including the USA, however, do not view CRISPR as that in the same light because it doesn't introduce foreign DNA into the organism, rather it just changes or mutates a gene.
All right, so for our first example, we look at a study that aimed to introduce the CRISPR-Cas9 ribonucleic proteins directly into the protoplast of Chardonnay grapes and Golden Delicious apples. They chose these two cultivars because they're commercially quite important. And they describe this as a DNA-free method because there's no transgenesis involved, so there's no foreign DNA being incorporated, and it wouldn't face the same harsh regulations as GMOs do. All right, so the issues at hand that they're trying to tackle are two diseases known to affect the crops. One is powdery mildew in grapes caused by a fungal pathogen, and the other is fire blight disease in apples caused by a bacterial pathogen. And the method involved targeted mutagenesis of certain target genes to increase resistance to the diseases. This included the MLO7 gene in grapevine, which is known for modulating pathogen defense, as well as DIPM1, 2, and 4 in apple, which physically interacts with the disease-specific genes from the bacterial pathogen, and the single guide RNA chosen for the method was selected for the maximum knockout efficiency. So the protoplasts were resuspended and transformed with differing Cas9 and single guide RNA ratios, and the highest mutational frequencies were observed in 3:1 for ML7, 3:1 for DIPM1, 1:1 and 3:1 for DIPM2, and 1:1 for DIPM4. And the next example, we turn to the meat industry, and we're looking at a study that aimed to develop a method for efficient one-step generation of mutant pigs by injecting CRISPR-Cas9 straight into the zygote. And in this case, we're looking at the VWF gene, which encodes the von Willebrand factor, and a deficiency of this protein results in von Willebrand disease, which causes an increasing bleeding tendency. And you might think that just has an application in like disease modeling, because this is also present in humans, but it also has one in agriculture, which I now will explain. So an important process in meat production is to bleed out meat, because any leftover blood can potentially cause growth of harmful bacteria. So the VWF gene is targeted so it's knocked out, which is thought to increase bleeding efficiency, and thus this would make the meat production safer, as there would be less leftover blood in the meat. So the single guide RNA that they chose targeted the exon 5 of VWF, and knockout of this sequence was thought to result in a loss of function. And the CRISPR-Cas9 was micro-injected into the zygotes, as can be seen by the figure. And 16 piglets were born. 2 of them unfortunately died shortly after birth. But with Sanger sequencing, they were able to tell that 3 of them were wild type, 5 were monoallelic mutants, and 6 were biallelic mutants. And the bleeding time of the biallelic mutant pigs was much longer than the wild type and monoallelic, meaning that they bled out more blood than the others. And aside from that result, they furthermore, CRISPR demonstrated that the process had little toxicity development to the embryo. No off-target effects were seen, and the mutations were transmitted through the germline.
We'll be looking at, in this study, it's looking at broad virus resistance in non-transgenic cucumbers. The aim of this was to cause viral resistance in cucumbers against cucumber vein yellowing virus, zucchini yellow mosaic virus, and papaya ring spot mosaic virus. The Cas9 sgRNA was used to target the C and the N terminus of the EIF4E gene. The cucumber yellow veining virus, vein yellowing virus causes clear veins, loss of chlorophyll, and often causes death of that part of the plant, where the zucchini yellow mosaic virus causes stunted fruit growth, causing deformation, and the papaya ring spot mosaic virus causes a reduced fruit abundance and affects the quality of the flavor. They are all proviruses, which need specific conditions to live. However, without the EIF4E wild type gene, those conditions can't be met, and the virus can't proliferate. There are 2 types of the EIF4E gene in cucumbers. It's the EIF4E and the EIF iso4E. Which are very similar. They have 50% nucleotide and 60% amino acid homology. However, in this study, only the EIF4E gene was used. This, it was 2 regions of the genome were targeted. The axon 1 and axon 3 were targeted by the sgRNA, and these were used because they did not have homology with the EIF iso4E. This was done with agrobacterium-mediated transformation of 5 individuals of the T0 line. In the T1 line, it was found that, they only decided to use further from the T1 line, the T1 generation. They wanted to use line 1 and line 4 of the 5 independent plants. line 1 was shown to be heterozygous for the mutation, while line 4 was shown to be homozygous for the mutation. They found that homozygous recessive mutants in the T3 line showed resistance to all the viruses, and the heterozygous mutants didn't show the same resistance. There are a large amount of benefits to genetically engineering our food. The health benefits are evident as crops have not lost quality from viruses and diseases. The ability to resist viruses and diseases and to even make larger, more abundant yields can help improve the income of the farms, but also for the country. With cheaper growth and easier ways of farming, healthy food can become more affordable and accessible. This becomes ever more important in developing countries where people suffer from numerous deficiencies and health complications. Health becomes the main concern. Sorry. In conclusion, I hope that this presentation shows that genetically engineering food isn't as bad as people might think it is. That research into CRISPR techniques has sparked really interesting ways of making DNA-free or non-transgenic plants and animals. And this has led to ways of making large-scale genetic manipulations as further generations do not have to be manipulated, and only the T0 lines have to be manipulated.